Yeah, good morning all. Today's topic is compound lipids, phospholipids, amphipathic lipids, and steroids. So, what are the objectives of this lecture? To learn about the compound nature of the lipids and phospholipids classification and functions, and the amphipathic nature of the lipids and regarding the biomembranes, Micelles, liposomes and emulsions, and also about the steroids. The complex lipids, otherwise called compound lipids. So what's the difference between the simple lipids and the compound lipids? We have already learnt about the simple lipids where the fatty acids are esterified to the glycerol or other alcohols. So there is no other extra group is there in the simple lipids. Whereas in complex lipids, they contain fatty acids esterified to the alcohol groups and along with additional groups are present. So here if you see the phosphatidylcholine and the sphingomyelin. So in phosphatidylcholine, the what are the extra groups? Phosphoric acid and a nitrogenous base, that is choline. So here the alcohol is the glycerol. So there are two fatty acids and if you see the sphingomyelin structure, then there is a the alcohol is the sphingosine and the fatty acid is only one, and there is a phosphoric acid as well as a nitrogenous base. So because of this extra group or additional group to the simple lipid, so that is making it a complex lipid. So what are these complex lipids? We'll see. So if we classify the compound lipids, so it is classified into phospholipids and non-phosphorylated lipids. So in phospholipids, again, nitrogen containing glycerophosphatides, that is phosphatidylcholine, otherwise called lecithin, and instead of choline, if there is ethanolamine nitrogen base, then it is called cephalin. Then the other one is phosphatidylserine. Then what about the non-nitrogen containing glycerophosphatides? So here it is a glycerophosphatide, but the base is a compound that is not having nitrogen, like inositol. So phosphatidyl inositol, then phosphatidyl glycerol, diphosphatidylglycerol, glycerol, also called as cardiolipin. Then third one is plasmalogens, which have long chain alcohol, that is either choline plasmalogen or ethanolamine plasmalogen. Then fourth one is phosphosphingosides with sphingosine alcohol, that is sphingomyelin. Then coming to non-phosphorylated lipids, here there is no phosphoric acid. So glycosphingolipids, which contains carbohydrate groups. So cerebrosides, that is ceramide monohexosides. Then globosides, ceramide oligosaccharides. Then gangliosides, that is ceramide plus oligosaccharide, that is globoside plus NANA. What is NANA? N-acetyl uramic acid. So these are glycosphingolipids which contain the carbohydrate component. So it is a combination of both carbohydrate and lipid. So I will show you the diagram later. Then coming to the sulfolipids, which contain the sulfated groups, are also called sulfatidides. Sulfated cerebrosides, sulfated globosides, sulfated gangliosides. So this is about the classification of the compound lipids, otherwise called complex lipids. Okay, don't get confused when given in the exam as complex lipids are separate and compound lipids are different. It's not like that. So coming to the most important essay question, short notes and viva notes and multiple choice question, phospholipids, otherwise in short form called PL. So they contain glycerol, fatty acids and a nitrogenous base. So L-phosphatidic acid is the simplest phospholipid. So what is this L-phosphatidic acid? We'll see later. So it was discovered in 1861, that is in the 19th century. So Straker characterized choline. In 1870, lecithin was discovered by Ernst Celler, who is a German biochemist. And in 1884, Thudicum JLW described sphingomyelin, cerebrosides, cephalin, and lecithin in brain tissue. Then we are coming to the phosphatides. So these are the derivatives of the phosphatidic acid, which is the simplest phospholipid. Then what is this phosphatidic acid? It is made up of one glycerol to which two fatty acid residues are esterified to carbon atoms, first and second one. So what about the third hydroxyl group? It is esterified to the phosphoric acid. So phosphoric acid esterified to the third hydroxyl group is called as phosphatidic acid. So this molecule has an asymmetric carbon atom and therefore exhibits a optical isomerism. So naturally the L isomerism is found for the phosphatides. So the most important character on which we survive is amphipathic nature of these phospholipids. So phospholipids are amphipathic in nature, particularly lecithin. So they have both hydrophobic and hydrophilic portions in their molecule. So the glycerol along with the phosphoric acid and choline constitute the polar head of the phospholipid molecule, whereas the hydrocarbon chains of the fatty acids represent the non-polar chain. So obviously you know that the polar means water soluble, non-polar means it is fat soluble. So the fatty acids represent the non-polar tails. So what are the most important amphipathic lipids? Lecithin, cephalin and sphingomyelin. So this is a viva question as well as a MCQ question. So don't forget. So plasma membrane, if you see the cell membrane, it is a phospholipid bilayer. So here you can see the hydrophobic non-polar tail uh, and the hydrophilic heads which face towards the water. So the tails usually orient away from the water, that is because of the hydrophobic force. This phospholipid forms a bilayer in the plasma membrane. So this diagram all of you please learn. So the molecules align themselves to form the monolayers with the polar heads pointing in one direction and the non-polar tails in the opposite direction. So only fatty acids with more than 6 carbon atoms form monolayers. This explains their role as components of the biomembranes. So the self-assembly of phospholipids into bilayers is driven by the hydrophobic interactions. They also act as detergents and emulsifying agents. We'll see what is emulsification in the later slides. So in vivo, they act as pulmonary surfactants. So coming to the most important phospholipid, that is phosphatidyl choline, otherwise called lecithin. So this is a nitrogen containing phospholipid. So the word lecithin is derived from the Greek word lekithos, which means egg yolk. So it contains the alcohol is glycerol. So the alpha and beta positions are esterified with the fatty acids, and usually this fatty acid attached to the beta carbon, that is the second carbon, is the polyunsaturated fatty acid molecule. The phosphoric acid is added to the third position to form the phosphatidic acid. The phosphate group is esterified to the quaternary nitrogen base called choline. The molecules of lecithin exist as deuterions with the isoelectric point as 6.7. So what is the action of the phospholipases? Here you can see in the diagram. So numbered as 1, 2 and 3 and 4. So number 1 is the site of action of the phospholipase A1. Whereas second number 2 is the site of action of the phospholipase A2, which is very important. And number 3 is the site of action of the phospholipase C. and number 4 is the phospholipase D. So the lysolecithin is a lysolecithin is formed how? So you might have known that snake bite is very poisonous, and it can lead to multiple organ failure, so which may lead to death. So what is there in venoms, vipers, snakes, venom poison? So that is phospholipase A2. So when phospholipase A2, which acts as the site 2, number 2, so it gives rise to, it releases a fatty acid, that is R2, and the rest of the compound is called lysolecithin. So what is the importance of this lysolecithin? It is a detergent as well as a hemolytic agent. So the enzyme present in the venom of the vipers is the phospholipase A2. So the hemolysis and consequent renal failure seen in viper poisoning could thus can be explained. So likewise, as I told, phospholipase A2, which breaks down the lecithin into lysolecithin and fatty acid, whereas phospholipase A1 breaks the lecithin into acylglycerol phosphorylcholine and releases a fatty acid. And phospholipase C breaks down the lecithin into 1,2-diacylglycerol and phosphorylcholine. And phospholipase D breaks down the lecithin to phosphatidic acid and choline. So here you can see phospholipase C is also important, especially in the signal transduction, which we will see later. Then coming to one of the most important clinically relevant biochemistry, that is pulmonary surfactants. The normal lung function depends on a constant supply of the lung surfactants, which is produced by the alveolar epithelial cells, and it decreases the surface tension of the aqueous layer of the lung and prevents collapse of the lung alveoli during end exhalation. So the surfactants seen at the air hydrophobic tail and water hydrophilic tail interface of the alveolus. So in this diagram you can see the alveolar cells of type 1 and type 2, and the air water surfactant layer spreading all over the alveoli endothelium, and you can see other parts of the alveolus. You can see this alveolus is a small size and spherical shape, and which can be easily collapsible. So this collapsibility is prevented by the action of the surfactants. So then what is the composition of the pulmonary surfactant? So dipalmityl phosphatidylcholine, which makes 40% of the composition of the pulmonary surfactant. Then other phospholipids make it to 40%, that is phosphatidylcholine 85% and phosphatidylglycerol 10%, and which are formed in the lamellar bodies in the alveolar cells. Then what about the other 5% by the surfactant associated proteins, also called SPA, SPB, SPC and SPD. What is SP? SP is surfactant associated protein, which makes 5% of the composition of the pulmonary surfactant. Then cholesterol makes another 5%, and the proteins like plasma proteins and apolipoproteins of surfactant proteins makes the rest of the 10%. Then what is how it is clinically relevant? So this the deficiency of surfactant can lead to respiratory distress syndrome, otherwise called RDS. So there are different types of RDS. So this is a one of the most common cause of the death in preterm neonates, which is due to the lower quantities of the surfactants. Why lower quantities of surfactant? Because immature lungs cannot synthesize the dipalmityl lecithin. So acute RDS, infection leads to injury to the endothelium, leads to the pulmonary edema, and meconium aspiration syndrome, which destroys the surfactant, and pulmonary alveolar proteinosis, also called as PAP, So there are inborn errors of sp metabolism. So all these conditions can lead to the surfactant deficiency, which can cause the collapse of the lungs, which leads to the respiratory distress syndrome. So there is another condition, neonatal hyaline membrane disease, neonatal RDS, or called infantile RDS. So this is also due to the deficiency of the surfactant. Then phosphosphingoside. So they contain phosphoric acid group. So a common phosphosphingoside present abundantly in biomembranes, especially on the nervous system, is sphingomyelin. So it contains choline. So sphingomyelins are the only sphingolipids that contain the phosphate and have no sugar moiety. So different sphingomyelins may be formed depending upon the fatty acid attached. So lignoceric acid, that is 24 carbons, and nervonic acid, that is 24 carbon with one double bond. Nervonic means the nervonic fatty acid is an unsaturated fatty acid because it has one double bond. And cervonic acid, which has 6 double bonds with 22 carbons. So these form different sphingomyelins. So because of its amphipathic nature, sphingomyelin can act as an emulsifying agent and detergent. And the relative proportion of lecithin and sphingomyelin is important in the biological fluids like bile, amniotic fluid, etc. So sphingosine combined with fatty acid is called ceramide, which is a component of the glycosphingolipids. So what is the clinical relevance of this lecithin and sphingomyelin? So LS ratio, that is lecithin to sphingomyelin ratio, and the presence of the phosphatidyl glycerol (Pg) correlate with the lung maturity. So during fetal life, the lung synthesizes sphingomyelin before 30th week of the gestation. So, but as the fetus matures, that is after the 30th week of gestation, so more and more of lecithin is produced. So this LS ratio of amniotic fluid is an index of the fetal maturity. So a ratio of 2:1 indicates a full lung maturity, whereas the lower levels indicates the prematurity. So these low levels, as I told previously, low levels of this surfactant can lead to the RDS, which is a common cause of the neonatal morbidity. Then apart from lecithin, what is the other phospholipid, that is phosphatidyl ethanolamine, or otherwise called cephalin. So cephalin differs from lecithin in that the nitrogen base is different, that is ethanolamine is present instead of the choline. So cephalin is also found in the biomembranes and possesses the amphipathic properties. Phosphatidyl inositol. So here the phosphatidic acid is esterified to the inositol. So phosphatidyl inositol 4,5 bisphosphate, or otherwise called PIP2, is present in the biomembranes. So this compound plays a vital role in the mediation of the hormone action on biomembranes and acts as a secondary messenger. So this is involved in the signal transduction of the both intracellular and extracellular components. So the PIP2 pathway, the muscarinic receptor of a GPCR, that is G protein coupled receptor, that stimulates an enzyme known as phospholipase C. So here in the diagram you can see the phospholipase C action at the site 3, which produces the intracellular mediators such as IP3, that is inositol triphosphate, and DAG, diacylglycerol. So these act as second messengers. Then coming to the plasmalogens, these are glycerophospholipids containing an alkynyl ether lipid. Alkynyl ether lipid, you can see in the diagram on the right side the ether linkage CH2O CH, and the alkene compound alkene bondage, that is CH to CH double bond CH. So the nitrogenous base may be either ethanolamine or choline. So these have an aliphatic long chain alpha beta unsaturated alcohol in ether linkage. with the first hydroxyl group of the glycerol. So the alcohols have about 12 carbons to 18 carbon chain length. So where these plasmalogens are found, they are found in the biomembranes of the brain and muscle. Then another phospholipid, that is phosphatidylglycerol, that is composition is phosphatidylglycerol is a complex lipid containing phosphatidic acid molecule esterified to the glycerol. Then diphosphatidylglycerol is found in many mitochondrial membranes. So it is also a component of the pulmonary surfactant, and it is also precursor for the biosynthesis of the cardiolipin. So what is this cardiolipin? It is diphosphatidylglycerol. So the decreased cardiolipin levels can lead to the mitochondrial dysfunction, and it is accounted for the heart failure, hypothyroidism, and some types of myopathies. Then what is APL antibody syndrome? That is anti-phospholipid antibody syndrome. So these anti-phospholipid antibodies are found in blood of patients with autoimmune disorders and in patients of thrombotic episodes. And also in pregnancy, so the presence of these anti-phospholipid antibodies can lead to abortions, and it can lead to eclampsias and also the premature labor. So here these antibodies are developed against the membranes phospholipid, that is, cardiolipin. Then sphingolipids, we have already learnt about the sphingomyelin to stress for the LS ratio. And here the sphingosine containing lipids may be of three types: phosphosphingosides, glycosphingolipids, and sulfatidyls. So all sphingolipids have the long aliphatic amino alcohol sphingosine. Here you can see in the first diagram, which is attached to a fatty acid in amide linkage to form a ceramide. So on the right side you can see the ceramide structure. What is a ceramide? It's nothing but sphingosine, which is a long aliphatic amino alcohol which is esterified to the fatty acid. So sphingosine plus fatty acid is a ceramide. So sphingomyelin structure, here you can see the sphingosine group which is esterified to the fatty acid, a single fatty acid you can see, and also it has got phosphoric acid along with the choline group. So this is the structure of this sphingomyelin. So whenever it is given in the examinations, please draw this diagram. Non-phosphorylated lipids. So glycosphingolipids or glycolipids, they are seen widely in nervous tissues, and this group of lipids do not contain a phosphoric acid, but instead of it, it contains carbohydrates and ceramide. So ceramide plus glucose. Ceramide, what is ceramide? It is a sphingosine with a fatty acid, and ceramide plus glucose is glucocerebroside, and ceramide plus galactose is galactocerebroside. And what is a globoside? It is a ceramide oligosaccharides, that is multiple hexoses. That is, they contain two or more hexoses or hexosamines attached to a ceramide molecule. So ceramide plus lactose is lactosyl ceramide, a component of the erythrocyte membrane. Gangliosides, they are formed when ceramide oligosaccharides have at least one molecule of N-acetyl nuraminic acid or sialic acid attached to them. So ceramide lactosyl ceramide with nana, they are designated as GM3, that is ganglioside M3. So ganglioside contribute to the stability of the paranodal junctions and ion channel clusters in myelinated nerve fibers. So autoantibodies to GM1 disrupt the lipid rafts, paranodal or nodal structures, and ion channel clusters in peripheral motor nerves. So it is seen in peripheral neuropathies, multifocal motor neuropathies, sensory neuropathies, and Guillain-Barré syndrome. So GM1 has an important physiological properties and impacts the neuronal plasticity and repair mechanisms and the release of the neurotrophins in the brain. So besides its function in the physiology of the brain, So GM1 acts as the site of binding of both chlorotoxin and E. coli heat level enterotoxin, that is traveler's diarrhea. So in this diagram you can see the structure of composition of the GM1, GM2 and GM3. So as I told, this yellow circle is showing ceramide, that is sphingosine with a fatty acid in the amide linkage. So GM3 is galactose and glucose attached to this ceramide along with the nana, and to this GM3, if N-acetyl galactosamine is attached, it is GM2. And what is GM1? So when beta D galactose is attached to the GM2, it is GM1. Then sulfolipids or sulfatides. These are formed when sulfate groups are attached to the ceramide oligosaccharides. So these complex lipids are important components of the membranes of the nervous tissue. Failure of the degradation of these compounds results in the accumulation of these complex lipids in the CNS. So this group of inborn errors is known as lipid storage diseases. Also, these are lysosomal storage disorders because the enzymes which are deficient are found in the lysosomes. So whenever there is a deficiency or defect in the breakdown of the sulfolipids, then this leads to the accumulation of these sulfolipids, which leads to the lysosomal storage disorder. So these are also known as sphingolipidosis. So for example, metachromatic leukodystrophy. So this is due to what deficiency of enzyme? It is sulfatase enzyme deficiency. So if this enzyme is deficient, then what is going to accumulate? It is sulfo-galactose erythrocyte is going to accumulate in the lysosomes. Then coming to the micellar formation and liposomes, you can see in this diagram the lipid bilayer, the polar groups and non-polar tails. Because of the hydrophobic interactions, they align themselves, and Micelles, they have aligned in a spherical format with an non-aqueous or hydrophobic center point. And the liposomes on the right side, you can see it is a the layer is the bilayer itself with an aqueous center point. So Micelles are molecular aggregates of amphipathic lipids, and which forms the biological membranes. Examples are formation by the phospholipids and bile salts. Which why this micellar formation is important? It's for the absorption of non-polar lipids in the small intestine. So absence of these micellar formation by the absence of the bile salts can lead to steatorrhea, that is a fatty diarrhea. Then what is emulsion? These are smaller droplets of non-polar lipids formed by the interaction of the non-polar lipids with the water or polar lipids. So emulsification of triglycerides by bile salt enhances the action of the pancreatic lipase. So by breaking down such a bigger fat globules, these bile salts enable them to form smaller emulsified droplets, so that the pancreatic lipase active site comes into play, which leads to the digestion of the fats. Then liposomes. So a lipid bilayer will close on itself under appropriate conditions to form the liposomes. So there are two types, unilamellar or multilamellar liposomes. So they may be prepared by sonification of mixtures of phospholipids and cholesterol. So these are microscopic spherical vesicles. When mixed in water under special conditions, the phospholipids arrange themselves to form a bilayer membrane which encloses some of the water in a phospholipid sphere. So drugs, proteins, enzymes, genes may be encapsulated by these liposomes, which could act as carriers for these substances to target organs. So the liposomes entrapped drugs exhibit superior pharmacological properties than those observed with conventional formulations. Liposomes have important applications in cancer chemotherapy, antimicrobial therapy, gene therapy, vaccines, and diagnostic imaging. Then coming to the last part, that is steroids. Steroids are compounds having a characteristic cyclopentanopenhydrophenanthrene ring structure. So sterols containing alpha hydroxy or keto group on carbon 3 and aliphatic side chain on carbon 7. So the most important of steroids is cholesterol. So the chemistry is cholesterol is a 27 carbon sterol. It has a hydroxyl group at the third carbon and a double bond between the fifth and sixth carbons, and eighth carbon side chain on carbon 17, and cholesterol forms cholesterol esters through its hydroxyl group on carbon 3. So cholesterol is exclusively found in animals. It's not a plant fat. In the plants, the counterpart of cholesterol is ergosterol. So it is mainly found in cell membranes and lipoproteins such as low density lipoproteins and high density lipoproteins. So compounds that can be derived from the cholesterol include the following. So cholesterol esters, they are formed either in plasma or tissues, and 7-dehydrocholesterol is a C27 steroid. It is formed in the skin. It is a precursor of the vitamin D. So it is a precursor hormone. 7-dehydrocholesterol is also called as a precursor hormone. Then estradiol is a 18 carbon steroid. It is formed in the gonads and placenta. It is a female sex hormone. It is required for the maintenance of the secondary sex characteristics and also pregnancy and regulation of the menstrual cycle. And progesterone is a 21 carbon steroid. It is formed in the gonads and placenta. It is a female sex hormone and is required for the maintenance of the pregnancy and regulation of the menstrual cycle. So in this diagram you can see the steroid nucleus, that is cyclopentanoperhydrophenanthrene ring, and the cholesterol structure and other glycocolate and aldosterone, progesterone, cortisol and testosterone, 17 beta estradiol and 7-dehydrocholesterol structures you can see. Then another important steroid hormone is testosterone. It is a 19 carbon steroid. It is formed in the gonads. It is a male sex hormone. It is required for the maintenance of the secondary sexual characteristics in the male and the regulation of the spermatogenesis. Aldosterone is a 21 carbon steroid. It is formed in the adrenal cortex and it regulates the sodium metabolism. Cortisol is a 21 carbon steroid. It is formed in the adrenal cortex. It regulates the carbohydrate metabolism. Bile acids are 24 carbon steroids. They include glycocolate, tarocolate, glycoquinodeoxycolate, and taroquinodeoxycolate. They are required for the absorption of the fats and fat-soluble vitamins. They solubilize cholesterol in the gallbladder, thus preventing the formation of the gallstones. Then reduced forms are C27 steroids. These include coprostenol, alpha form, and cholestanol, that is beta form. They are the degradation products of the cholesterol produced in the intestinal lumen. Then what are the properties? Cholesterol appears as a yellowish crystal and the crystals show notched appearance when examined under the microscope. It is insoluble in water but soluble in organic solvents such as ether or chloroform. And what are the reactions of cholesterol? Cholesterol forms a blue colored complex when it reacts with acetic anhydride and concentrated H2SO4, that is sulfuric acid. So this reaction is called Lieberman-Bouchard reaction. It is used to determine the concentration of cholesterol in the plasma. So this finishes our topic, compound lipids.